Hello everyone, I'm Mingbo Cheng, currently a PhD student from Institute for Computational Genomics. Today, I present MOJITOO, a method to integrate multimodal data. With the development of single-cell sequencing techniques, nowadays, we are able to simultaneously measure two modalities at the same cell, like simultaneously measure transcriptome and genome, or with proteome or epigenome. We can even measure more than 3 modalities at the same cell. For example, T sequencing enables us to measure 3 modalities, and the dogma sequencing can even measure 4 types of features. For multimodal data, here RNA, ataxic, and ADT modalities, they share the same barcodes, that is, they share the same columns. But the feature space of these modalities are really different. The task of integration is to find a uniform latent space to embed all these features. With which, we can further run, for example, UMAP to visualize, or to perform clustering, or to do the trajectory analysis, or dig into gene regulatory network. There are several challenges for the single-cell multimodal integration. First, different modalities hold distinct statistical properties. For example, feature sets of protein is around 100. A toxic feature space set is 100,000. And the gene expression matrix feature space is tens of thousands. And the sparsity of different modalities are really different. The general properties of protein, ataxic, and RNA are really different. Second, the integration method should be efficient enough so as to cope with increasing library size. Nowadays, we can sequence as many as even millions of cells. Third, more and more protocols are created to simultaneously measure features in the same single cell like open chromatin, gene expression, protein, DNA methylation, etc. And the combination of modalities types pose a challenge to the integration. Some method can only tackle with 2 modalities, and some others can work on specific feature matrix. Another aspect is the interpretation of its component of the latent space. When associate molecule features with the latent space, we can interpret its component using features from specific modality. So here I list 6 methods for integration. MOFA use matrix decomposition, and Schema use matrix learning. While WN create a weighted nearest neighbor network. Diablo use partial least square discriminant analysis. SAI also use matrix decomposition, while Liger use non-negative matrix factorization. In terms of the protocol these methods support, the top 4 methods can work on any kinds of modalities, while the bottom 2 can work on specific modality combinations. When it comes to the number of modalities that a method can work with, some methods only work on 2 modalities, and some others can work on any numbers of modalities. The efficiency is also one important aspect. Here we can see MOFA, Diablo, and SAA are too slow. It is a challenge for them to integrate large datasets. For interpretability, Schema use quadratic program, which distort the primary modality information from the others, so that the association to features of each modality is not interpretable. Well, WN only provide a network rather than the latent space, so it's not interpretable as well. For all these methods, we have to set at least two parameters as prior knowledge. Namely, we have to add assumptions before run these methods. Overall, every method has its shortcomings. So we propose MOJITOO, which is based on canonical correlation analysis. It is universal, can work on any kinds of modalities. And it is a simple method, really fast to run. And it is also interpretable. On top of that, it is parameter free, meaning no prior assumption for the input. The idea of MOJITOO is using canonical correlation analysis. Here we show geometrical interpretation of CCA. Taking RA and ataxic, for example, the inputs are RA and ataxic dimension reductions. As we can see, cell A and cell B, they have different coordinates in RA and ataxic space. CCA is to find a transformation to minimize the angle between zeta RA and zeta ataxic. We add up sensing components of a cell from RA and ataxic as the MOJITOO coordinate in CCA space. The detail of MOJITOO workflow is as follows. If there are two modalities, for example, RA and ataxic modalities, with the dimension reduction, the running would be more efficient without losing information. We take these as input of MOJITOO and run CCA to get a sensing component from RA and ataxic. Before summing up, MOJITOO denoise the signal by keeping only significant correlated CC component. If there are more than two modalities, for example, if there is another protein modality, MOJITOO first arrange the modalities by number of features from larger to smaller, and next to run CCA iteratively to get the final MOJITOO latent space. We use 6 publicly published datasets to perform the benchmarking. As you can see, the first two use set sequencing protocol, where two modalities are RNA and protein. And the third and the fourth datasets involve RNA and toxic modalities. And the last two datasets include 3 modalities. We compare MOJITOO with 7 methods: schema, WN, SAI, Liger, MOVA, Diablo, Symphony, integration. To note some inputs of this method, we use dimension reduction to speed up the running without reducing the performance of this method. For schema, SAI, and Liger, they use their own strategies, so we didn't change their inputs. We use 5 metrics to do the benchmarking. The top 3 are to benchmark the accuracy of competing methods. Here, structure score is to measure the preservation of latent space, while silhouette score is to measure the distance representation, while adjusted random index measure the accuracy of clustering on latent space. And here, time elapsed and the memory usage are also compared. For the sake of time saving, I will only go into detail of the structure score, time elapsed, and memory usage. The preservation of shared latent space structure score for each modality is calculated by first obtaining the distance matrix of each modality. Take a toxic modality, for example, we can see the distance matrix up triangle entries is pairwise distance for all cells. The structure score is to calculate the Pearson correlation of flattened up triangle of the 2 distance matrix. We can obtain structure score for each modality. we finally average structure score across all modalities. Here, as we can see, MOJITOO has the best structure score in 4 datasets. Well, although BMSET and PBMC Dogma, MOJITOO score is smaller than MOFA and Diablo. Well, the variation of MOFA and Diablo are both higher than MOJITOO. This indicates MOFA and Diablo use more information from some modalities, while MOJITOO denoise the signal by extracting most common features from all modalities. So, a quick glance on the ranking of top 3 metrics. For the latent space preservation, MOJITOO performs the best, and the distance measure silhouette score, it is the second after MOFA dimension reduction. The clustering matrix adjusted rand index, MOJITOO is also the best. For time elapse and memory usage, we use skin data who has the largest number of cells, and we perform downsampling from 30,000 to 3,000. As we can see, MOJITOO needs least time faster than all competing methods. In terms of memory usage, MOJITOO is also the best of all. Here, please note that at least also the MOFA original version, which is not as efficient as dimension reduction version. Here, we expect each CC in datasets PBMC multimodal data. The MOJITOO component can represent the topic of cell types from both positive and negative. Here, high values of CC1 are associated to myeloid cells, while negative values are associated to T cells and NK cells. CCs are also capable to capture subtle changes between major cell subtypes. Here, CC4 captures changes between naive and activated T cells, while CC5 captures the naive and activated monocytes. If we multiply original feature matrix with MOJITOO component, we can also explore feature by CC matrix, like here gene by CC matrix and peak by CC matrix. And the top features in each CC are cell type specific. For example, here the heat map shows the top 10 genes, both positive and negative. Of CC1, we order the cells by CC scores. Here the expression of genes with high CC1 values include monocytes genes, like these 2 genes, while negative CC1 values are associated to T cell genes, like these 2 genes. We also observed that top-ranked peaks with high or low CC1 scores have monocytes or T cell specific open chromatin. To wrap up, MOJITOO we propose is a parameter-free method, and it is performed best for ARI structure score. It is the second after MOFA on silhouette score. MOJITOO ran faster and more efficient than all competing methods. And the latent components of MOJITOO capture major cell types. It is also capable to capture minor cell type changes. Top-ranked positive and negative features of CC are cell type specific. Finally, I would like to thank my supervisor Ivan Costa and my colleague Li for the work of MOJITOO. And also thank to thank my colleagues in my group. Thanks. I'm happy to answer your questions. MOJITOO package is available on GitHub. You are welcome to try the package and please contact me if you can encounter any issues.